Hey everybody, I'm going to go through the part two of the evolutionary forces and population notes. I actually made this video already once today at school. It took me about 40 minutes, and then when I got done, it had the whole bad lip sync thing going on. So I'm going to try and make another one here tonight so that you guys can hopefully understand things a little bit better. Last week I made some at school and they worked fine, so I don't know why sometimes it does and sometimes it doesn't, but it's getting very frustrating. So I know it's tough on you guys too, but I'm spending a lot of time redoing these guys, so uh, not too much fun. But here we go. So at least I think we're going. It won't pull up the slideshow. Hang on one second. Here we go. All right, sorry about that. So big questions for this unit. Is it possible to measure evolution? That's kind of a weird question, but we're going to be diving into it and see about how we might go about measuring evolution, and then what causes the variation in the populations, and how would measuring populations help us to understand things. So um, the first example here, I'm just going to kind of go through kind of quickly because this is all still basic natural selection stuff. It really doesn't even, need to be in here again. You guys have, I think you kind of get natural selection by now. We've done a lot of examples. So, survival is variable, reproduction is variable, populations evolve, fitness increases, for all of these traits that become, transferred over time. Okay. So, kind of a typical, experiment how depending on the rock color, the mice are evolving depending on the rock color and they're changing, based on the color. So the light mice are blending in with the light rocks and the dark mice are blending in with the dark rocks. And, the two in the bottom pictures don't blend in really at all. So they don't stand as good of a chance to survive and pass on their traits. So, um, the raw material of evolution is what we say is variation, right? And so there's a couple ways we get variation. There's actually gonna be more than two, but for now we're gonna talk about two. On the genetic side of things, remember we're kind of introducing genetics into things now with the modern synthesis, the neo-Darwinism stuff that we talked about in the first part of the notes with Haldane, Sewell, and Wright, those three scientists that kind of advance things with genetics. So, um, you know, their mutation is definitely going to be a way to get variation, right? Random changes to DNA. And why do those happen? Because, sometimes the mechanisms that copy DNA don't always work. That's the simple answer. We'll get to more of that in genetics and really look at why things like that happen. But that's not really, for now. So sexual reproduction is obviously another way we get variations. Why siblings are similar but not always the same, or not usually ever the same unless there's twins involved. So each offspring is a different arrangement of the DNA from the parents. Required terminology. I'm pretty sure this is going to be review for all of you. You probably saw this in middle school, even doing Punnett squares and stuff like that. So genes, determine traits. So eye color would be, like a gene, and the variant of the gene is the allele, and that would be like blue or brown. Okay, so the trait is kind of the big category is the gene, and then the variance of that trait um, is the allele. And we're going to talk a lot about alleles later today. Um, so all sexually reproducing organisms have two alleles for each trait. Dominant and recessive, I'm pretty sure you guys remember that, right? And they used to be shown in Punnett squares with like big B, little b, and so dominant traits, will show all the time and they will hide the recessive traits unless there's some, special circumstances going on. And there are special circumstances that happen, but we'll just kind of, treat things normally today. And then homozygous, heterozygous. Homozygous meaning the alleles are the same. Heterozygous meaning things are different, right? So homozygous dominant would be big B, big B. Homozygous recessive would be little b. And then your heterozygous would have one of each. Okay, so hopefully all of that is kind of review and it makes some sense to you. A few more terms here: population and gene pool. Population, a localized group of interbreeding individuals. Remember, so like, buffalo in Montana are not really in the same population as Buffalo in Utah or whatever other state they might be in, even Missouri. Um, they have to be together. They have to be able to interbreed to be in the same population. Okay. And then the gene pool is all of the alleles in that population, all of the big letters and little letters for all of the genes in that population is the gene pool. Okay. So, and then evolution, just kind of another way to say it, change in allele frequency in a population, right? Because allele frequencies are going to be what drive kind of the changes that go through. So change in allele frequency in a population over time. That's pretty much what we're talking about with evolution. I'm just going to kind of run through this quick. Again, just another experiment, that I don't think will be too surprising to you, but might want to get my pen to show you a couple things though. Um, so over here in this pool, we have a predator that's called a killifish, and he likes small guppies. And uh, then we have a predator down here in the bottom pool that's called a pike cichlid, and they prey on larger guppies. Okay, and so what they're going to do is pools with up here in this top pool up here now, pools that have killifish but not guppies prior to transplant. Okay, so they're going to transplant some guppies up there uh, into that um, into that pool. Okay, so since the pike cichlids prey on the larger guppies, there's some smaller ones here. Um, so got to get rid of this before we click over. And then here's kind of the data. So after 11 years, the average size and age of the maturity in the guppies in the transplanted populations increased compared to those in the control populations, right? So the one that like the small guppies um, uh, uh, would, it didn't, you want, sorry, hang on, got tongue tied. So the one pool with the killifish where they would have liked the small guppies, and you move some up there, the guppies ended up getting bigger and bigger to kind of avoid that predator. And you can see in the graphs how the sizes of the guppies increased, right? And so this is just another way, if you read through the conclusion, it's another way we were actually able to observe evolution. They spent 11 years looking at these fish pools, and so they were actually able to observe evolution in a pretty short time, 11 years, given that most things take hundreds or thousands or millions. In 11 years, the guppies change size pretty significantly. All right, so we have to look at 5 sources of evolution. So here's some more ways that variation can happen. We went over a couple. What causes variation? We talked about mutation. Gene flow, we talked about yesterday. Remember the flow of genes? That was with the deer example, and how things get more and more equal. They get more the same. You have non-random selection, sexual selection. Okay, remember sexual selection is not random. There are choices made. Okay, genetic drift. Remember that's bottleneck and founders. So that's going to play a role. And then we have natural selection, right? Then you have your typical natural selection, and there's variation caused by all of these things. Okay, so real quick, we'll run through them one by one. So with a mutation, the mutation occurs in the DNA, and then the amino acid sequence changes, and then the protein sequence changes, and that could very well bring about a structure and function change of a protein, and then possibly change the fitness of an organism. And so you can get change because of a mutation. With gene flow, okay, remember here we're talking about immigration and emigration and things moving into and out of a population. And so the genes are moving between the populations. And if that keeps happening, a lot of times things get more and more similar or there's less differences, right? And so gene flow has been increasing in humans over the past, let's call it 50, 60, 70 years. Is gene flow's really been happening quite a bit, and it really goes back to, if you want to think about it for a minute, go ahead, pause the video. It really goes back to um, commercial airlines being able to go around the globe at a much more prevalent rate, where there's lots more flights, transatlantic flights and transcontinental flights, and it moves people around all over the world. And some people end up staying there, and people end up, mating with other people around the world. And so there's been a lot of gene flow going on in the past 50, 60, 70 years. And so really it's happened because travel's been much more affordable and more people can afford to go around the world. Sexual selection, right? The traits that get you mates, right? And remember, there was inter and intra sexual selection. Um, and so there's choice, right? For the most part, there's choice. It's not random. And so another way we get variation is just through reproduction. Funny story about the beetles there at the bottom of the page. Generally, they would like want to fight it out with each other. Um, but what typically happens is they kind of go up to each other and they take each other's measurements. And whoever has the longest horns and the biggest body, they know eventually that one's going to win. So then the one that ends up being smaller just kind of turns around and walks away. Not even worth his time to fight. Why would I want to spend all that time and lose? So he just turns around and leaves and lets the bigger one have the female. So kind of funny. Genetic drift. So again, founders and bottleneck. Sorry about that. These a lot of times reduce variation, right? Because you lose a segment of the population, or you lose a bunch of the alleles because of the events that take place. The example to the right over here with our frogs, we have three colors in the top picture with a full pond, and we have yellow, dark green, and striped frogs. Then we get a drought. Okay, so it's going to cause a bottleneck because we get a drought and the pool level goes lower and there's probably less food and stuff like that, and so we lose the dark green phenotype is lost, and then the pond refills, but the only problem is the green frog does not come back, right? The alleles are gone at that point and they don't return, so we're still left with just the two colors. Okay. And then natural selection, you guys are, hopefully pretty familiar with by now. I think it's kind of an interesting way to phrase it up here at the top. Differential survival and reproduction due to changing environmental conditions. It's actually, pretty accurate. It's just a different way to say it. Things survive at a different rate based on what's going on and how their environmental conditions are favorable or unfavorable. So it's a pretty interesting way to phrase it, but I kind of like it. Um, and so obviously you can get a combination of alleles that increase fitness, and a lot of times things are passed on and things are improving, but not all the time. And so adaptive evolutionary change is also a factor. So things are adapting. So here's a question for you, and the question is, what conditions would cause allele frequencies not to change? Okay. What would keep allele frequencies the same? So what that question's really asking is, what would keep things from evolving? So pause the video for a minute or stop and think about it. Why would things not evolve? How would that happen? What sort of conditions would you need for things not to evolve? Okay. So hopefully we thought about or pause the video a little bit. First of all, couldn't have mutation, right? Couldn't have that. You couldn't have migration between populations, so no gene flow. Members of the population would just mate randomly, right? There would be no choice, there would be no sexual selection. The population is so large that genetic drift wouldn't matter, that you would have a natural disaster and it wiped out things. The population's so big it really wouldn't matter. You would just kind of keep going with the same allele frequencies. We wouldn't lose anything. And then no survival or reproductive advantage occurs, which means no natural selection, right? So in order to keep allele frequencies the same, these things can't happen, right? The problem is they all happen. We just went through this a few slides ago that these all happen. And so the question seems a little bit bogus, right? Of course these happen. And so why would we even go down this road of them not happening? So it's because we can use it to our advantage. Okay, we can use this to our advantage. So hang on one second. I gotta pause the video real quick. Okay, sorry about that. All set. So the Hardy-Weinberg equations, they describe a non-evolving population. They describe a population that is maintaining its allele frequencies. And it really serves as what we like to call a null hypothesis. If you've had statistics, you've probably heard about a null. If you took pre-AP bio, you probably do chi-square problems. At least we do at South. And you probably would have heard a null hypothesis. A null hypothesis is really what would happen if there was really no significant change. It kind of serves as um, a baseline, if you will. And so you're going to assume that there is no difference with the null hypothesis. And so it's, again, it's kind of a baseline for things that wouldn't happen. And then you kind of work against that statement. You either, with your data then and with your experiment, you either confirm um, or you accept or you reject is a better way to say it. You accept or you reject the null hypothesis that there is no difference. So if you accept it, then the data really showed you there was no difference in these things, and so there's really no change happening. If you reject the null hypothesis, then that means there is something going on. Okay, so um, natural populations are never in Hardy-Weinberg. Okay, they're never in Hardy-Weinberg equilibrium. Why would that be? Because those five things happen, right? Because mutations happen and gene flow happens and natural selection happens and all the things in that list and all the things in the slides we've already talked about because they do happen. So populations are not in Hardy-Weinberg. So again, why would we use this? Because it's a way to measure. It's a way to figure out if things are changing and how fast. So it's a model basically to see if forces are acting on a population. If there's no forces acting on a population, then the alleles will stay constant. If there's forces acting on a population, then the alleles will change over time. Okay. All right. So here's a little bit about the Hardy-Weinberg. Okay. So this is where we're going to talk about alleles. Remember, alleles are individual letters, right? So in this case, you might have a big B and a little b, right? So the frequency of the dominant allele would be a big B, and the recessive allele would be a little b. Rather than changing letters all the time with all these different problems, the Hardy-Weinberg equations, they always just use P and Q. Okay, so everything will just be P and Q, and um, P will stand for the dominant allele, and Q will stand for the recessive. So frequencies must add to be 100% because you can only have two alleles, right? And so if you have P and Q and those are the only two things you can have, then the two of them added up must be 100%, right? Because you're going to have some of one allele and some of another allele. And if those are the only two you can have, it has to equal 100% or 1. Okay. So we say P plus Q equals 1. That's the first equation we're going to deal with. So now, those are just alleles, but in order to show traits, right, and to have like one from mom and one from dad being passed on, we all end up with two alleles, right? And so if you are homozygous dominant, you would have two Ps, right? You would have two letter Ps, two dominant alleles. And so we call that P squared because you have two of them. Likewise, recessive, homozygous recessive would be Q squared, right? Because you have two Qs, so you're now Q squared. If you're if you're heterozygous, remember heterozygous is having one of each, but you could get the P from mom and the Q from dad, or vice versa. And so there's two ways to be P and Q. So we call that 2PQ because there's two ways to be that. So now there's only three things you can be, right? There's only three genotypes you can be. You can be homozygous dominant, you can be homozygous recessive, or you can be heterozygous. That's it. And so those three must also add to be 1. So P squared, dominant, 2PQ, heterozygous, Q squared, recessive, must also equal 1. So we have these two equations, both with P's and Q's. So the convenient part of that is once you know one letter, like if you know P, you can substitute that number in for the other equation. You can use these equations together simultaneously, and you can insert numbers. If you know one value, you can figure out the others for the other equation, and that makes things very useful. So I'm actually going to skip this. It doesn't tell us a whole lot, so I'm going to move on. This is a better slide. So let me bring up a pen here. So here's a couple of important things. And part of the issue with the Hardy-Weinberg is knowing which equation to use. Okay, so we went over these two equations right a second ago, but you got to figure out what they're used for. And if you use them for the wrong thing, you're just simply going to get the wrong answer. Okay, the math we're going to do is super simple. It's really just can you assign the right value to the right variable. Okay, that's really what we need to be able to do. So in this case, it says the gene pool is going to use this equation. Okay, gene pool is going to equal allele. Okay, so if you see a problem, if you see a problem talking about alleles, okay, then you need to use the gene pool or this equation. Okay, so make sure you're using, um, remember, alleles are single letters, right? Allele, big B, big T, little t. Those are single letters. Look at the equation. These are single letters. Okay, so that's why you use this equation for alleles. Okay, if you go down here, this says individuals. Okay, individuals can mean a couple different things. Okay, this might mean genotype, right? Okay, geno, which would be things like big T, little t, right? Two pairs of letters. And here there's two letters, right? Sorry, that doesn't, if I stop writing, it goes away. But genotype, big T, little t, okay? Um, big T, little t, heterozygous, doesn't matter. But there's two of them, okay? So all of these include two letters, okay? So anytime you see genotype or a phenotype, either one, okay? Because a phenotype requires two. My handwriting's not good anyway, and it's really bad with a mouse, so I apologize. But anytime you see a phenotype, okay, it requires two letters to make that phenotype, right? If I'm big T, and I'm tall, okay, it took two letters to make that phenotype. So anytime you see genotype, phenotype, individuals, appearance, any words like that, you're going to use this equation. And again, if you see alleles, single letters, you're going to use this equation. Okay, so it's super important in order to know which equation to use. So if we look at this, don't spend too much time on this. It's really just showing you why things work the way they do as far as mathematically. Why do they all equal 1? So if I were to show you real quick, we'll just pick the one in the middle here. So this says P and Q are both 0.5, okay? But the colored lines up here are the ones with the double letters, right? So here's Q squared or homozygous dominant, here's heterozygous, and here's homozygous recessive. So in this case, if you look right here, both the red and the blue line, okay, are basically at 25%, right? So you have your dominant and your recessive are both 25%, which means the third line, this one here, has to be 50% because they have to equal 1, right? So anywhere on this graph, if you were to look at this point and this point and this point, those three values would all add to be 1. Okay, and we could do this vertically at any point along here. Those three values would add to be 1. That's all it's trying to show you. Don't spend too much time on that. It's just validating those formulas. So how to solve these problems, right? So write down both equations, or at least have them handy, right? Just write them down once and keep them handy in a notebook or piece of paper or something. Identify the givens. These are going to be word problems. So you have to figure out what piece of information they're giving you. They have to tell you something. They have to give you one of the variables. You just have to figure out which one. Be aware of squaring frequencies. This is always really weird for people that aren't used to squaring frequencies. Squaring decimals is what we're really going to be working with, because they're going to give you percents and we're going to convert those into decimals when we do the math. So, for example, what is the square root of 81? Hopefully you can all figure out that the square root of 81 is 9, right? A much smaller number, okay? But what's the square root of 0.81? Stop and think about that for a second. Well, the square root of 0.81 is 0.9. That is a bigger number, right? So that seems very weird to us, but that happens with decimals, okay? So you're not doing it wrong. That's just kind of what happens. And I will tell you that some of the problems you'll do, they seem like they're very simple square roots because they look like normal numbers of square roots. Sometimes you might just want to double check on a calculator because it might be something just a little weirder than you're anticipating, okay? If you can find the letter Q, the single letter Q, the allele Q, the recessive Q, then that is really the magic variable that unlocks the others. And you might say, well, why not P? Because, P plus Q equals 1. Whether I find Q or P, it's kind of the same difference. The reason Q is better is because it's recessive, right? And because there's only one way to be recessive. So if you find the dominant one, sometimes it can still leave you with some questions about what the other things really are. Where Q is, since it's recessive, it kind of takes away the doubt. Doesn't mean it's always going to be possible, but if you have the choice, find Q. Work your way around the problem, solving for all the other variables. So once we know Q, think about this for a second. And I'll try and write again a little bit here. Once we know Q, right? So let's look at this formula. Oh gosh. P plus Q equals 1. So if I know Q and Q happens to be 0.4, right? Well, then then I automatically know P is 0.6, right? Because they have to equal 1. 0.4 and 0.6 are going to equal 1. So if I know Q, I automatically know P. But if I still know Q and Q is 0.4, like I said a second ago, I could also just say, well, I know Q squared, right? In the other equation, 0.4 squared, okay, is going to be 0.16. So you can go a couple different ways depending on what the question's asking you. So once you find one value, then you can kind of just work your way around the equation and start solving. If you know Q, you can find, P, and then if you know P, you can find P squared, and you already know Q, so you know Q squared, and you can take 2 times P times Q to get 2PQ. So it's pretty easy to work the math out around these problems. It's really just making sure you assign the right value to the right equation. Okay? Practice makes perfect, which we're going to do right now. So if 64% of the individuals in a population exhibit the recessive appearance, what percent of the gene pool is dominant? So that is our question. Okay? Go ahead and pause the video. Seriously, pause it and try and work this one out. Okay? And see how far you can get. And then go ahead and unpause it and see if you can match up what I'm about to do here. But pause the video now. Do it. Make sure you practice. All right, so let's take a look at this. So they exhibit the recessive appearance, right? 64% exhibit the recessive appearance. So that's the only number we have is 64%. Okay. So the question is, what variable does 0.64 equal? We have to figure this out to begin with. Okay. So we know it's recessive, so it has to really either be one of two things, right? It's either going to be Q or it's going to be Q squared, right? So is the recessive appearance, is that talking about an allele? Is that talking about a single letter? Or is that talking about something like genotype or phenotype, or like it even says the word in here, the individual? And then one of the equations said individuals, right? If you click back, you'll see that. So 0.64 should equal Q squared. And I'm going to have to work quickly here and keep drawing, or else it's going to kick me off and you won't be able to see the rest. So if I know Q squared is 0.64, then I also know Q, by taking the square root, is 0.8. And if I know Q is 0.8, then that must mean P is 0.2, right? And so what percent of the gene pool is dominant? What is this asking? What is gene pool asking? Well, that was the top equation, the one that actually said gene pool. And that was the one where I put allele. So that's the single letter, and it was asking for the dominant one. So it's asking for P, and that's that last one I just found, I believe, right? So it should be your answer is 0.2. Okay, for this problem. And they didn't ask us anything else, so we really didn't need those other variables, but we could have found them if we wanted to. Okay, so let's try again. Okay, pause the video, give this problem a whirl, and then kick it back on, and let's see how you did. Okay, go ahead. All right, so hopefully you did that, and you're not just watching me do some math problems here. So a population contains individuals, 16%, whom show the recessive trait. Again, this is the only number we have, 16%. It shows the recessive trait. So again, it has to be Q or Q squared because it's the recessive trait. And the individuals are showing this, okay? So what that really means is that 0.16 is once again q squared. We're dealing with two numbers, so 0.16 is going to be q squared. So now, what percent of the population is pure dominant? If it's pure dominant, that means two p's. So we need to find what p squared is, okay, for this one. What percent of the gene pool is recessive? So that is going to be Q, right? The allele for recessive is going to be Q. And what percent of this is hybrid? And hybrid means a mix, right? So that means you're looking for the 2PQ. It means you're looking for the heterozygous one. So really we kind of need everything, right? So again, sorry about my decimal being way up top. 0.16 is going to show the recessive trait. So that is going to be Q squared, which now means Q, okay, is going to be 0.4, which now means P is going to be 0.6, and that was one of your answers, and I think the other one was as well. And then the hybrid, sorry, I just let it go, but the 2PQ, well, P and Q were 0.6 and 0.4, right? So 2 times 0.6 times 0.4. 0.4 times 0.6 is 0.24 times 2. So our 2PQ is going to be 0.48, right? So we made it to 48% is going to be the hybrid. Sorry I do that a little quick. I just kind of wanted to keep the drawing on the screen for you. You can always pause it and look at it. So I think we solved every one of those questions it asked for, and you're going to be doing some more of these as practice, okay? So the last question really isn't a math problem at all. How can you identify if a population is in Hardy-Weinberg equilibrium? You measure it, right? You go down to a local pond, you measure the alleles or colors of particular frog species, and you write down your allele frequencies, and then you check it. You check it maybe every 6 months, okay? And you see what the allele frequencies are when you go back. And if those allele frequencies are exactly the same every time, then there's no changes happening at that moment. There's no evolution happening because Hardy-Weinberg is telling you that the allele frequencies are not changing. Now, if you go back and the allele frequencies are changing, then that means there are forces working on that population of frogs. And rather than everything being 50-50 at the beginning, now it's 60-40, and then it's 70-30, and the alleles are changing, then some or more, one or more of those five things, mutation, natural selection, gene flow, genetic drift, sexual selection, one or more of those things is happening to that population, and that's what scientists would then do. They'd go figure out, well, why is this population changing? Is it changing for the good? Is it struggling? Is it doing okay? And so that's really what we use Hardy-Weinberg for, is to figure out how populations are doing and if they're changing over time, okay? So a little quick application here. A lot of you guys have heard about sickle cell. Thought there was a picture. And so sickle cell is when you have these, crescent moon shaped red blood cells, right? And so due to a mutation in coding for the hemoglobin, oxygen carrying blood protein, right? Hemoglobin binds the oxygen to our red blood cells. If you are dominant, then you are big H. If you are recessive, then you are little h. So you have to be little h in order to have the disease, right? In order to have sickle cell, you have to be little h. If you have even one big H, then you will be fine. You will not have sickle cell. Symptoms down here below, lots of problems. Low oxygen, so the blood sickles. It looks like a crescent moon. Breakdown of red blood cells. The crescent moon shaped things will form clots, which is not great. Could cause organ damage and can be lethal in children. But here's the thing. If you have sickle cell in Africa, it might not be a terrible thing. This is why. Not only is sickle cell a thing in Africa, but so is malaria. Malaria kills a lot of people in Africa. And these two things work very differently. So heterozygous frequency is really high for such a lethal disease. So about 1 in 5 people are big H, little h in Africa, and that's a pretty high percent. And so If you are homozygous dominant, if you are big H, malaria is transferred through the blood. It likes your big, juicy red blood cells, right? Mosquitoes move it from person to person, and your blood gets infected. And so, um, if you have the nice, big, fat, juicy red blood cells and you get malaria, good chance you could die. If you have homozygous recessive and you have sickle cell, there's a good chance you could die from sickle cell. So being one of those two things is not great, right? You're going to have reduced reproduction, so then the frequency of those is going to go down. If you are heterozygous, you are partially immune to both malaria and sickle cell. You have enough plump red blood cells to feed your body the oxygen that it needs. Um, and that will also mean you don't have too big of an issue with sickle cell. If you have partial sickle cell because you have the little h, then you will you might get malaria, you might get a little sick, but generally you don't die from it because you have enough sickle cells that the malaria doesn't like, and so you don't get as sick as other people. And so then big H, little h becomes more common over there, and it's what's called the heterozygote advantage, okay, where it's an advantage to be the heterozygous one. So pretty interesting thing over there, and how that all works. So there you go. There's some practice questions, and hopefully you did okay with the Hardy-Weinberg stuff. There's plenty of stuff on the internet. If you didn't quite catch on, we are going to do a bunch of practice. The first, set of problems I'm going to give you won't even be graded; they'll just be practice. So we'll work on it some more. I think a lot of you have seen it before. You should have seen it pre-AP. So some of you will probably, blow through this, and it won't be too big a deal. If you've never seen this, then it does take a few minutes, take some practice to catch on for sure. Okay. Well, hopefully this helped, and hopefully I don't have to do this one for a third time.